Hello, in this video we will discuss about the T cell activation and proliferation. We will also understand about the HIV infection and why the HIV is unable to proliferate and unable to combat with the HIV virus. So let's begin to understand. First is the death associated molecular pattern and death associated molecular pattern and pathogen associated molecular pattern bind with a PRR, and in this way you can see the T cell basically is the CD4 T cell. You can see this is bind with the CD4 molecule and the T cell receptor with MSC class 2, which represent the part of the antigen as a epitope. It is the peptide we recognize and bind with the CD40 and CD40 binder with the bind, and in this way the produce. The third one is the activation of the dendritic cell. Activated dendritic cell produce the interleukin 12. This bind with the interleukin receptor. So in this way you can see, and in this way the CD4 T cell is the helper T cell, is used for the recruitment of the some other lymphocyte for killing and the inflammation side, for example. So here the signal 1 is the antigen specific interaction, and the signal 2 is the co-stimulatory molecule, and the signal 3 is the intrusive cytokines production, which that is basically complete activation of the T cell, CD4 T cell, which that is used for the activation and recruitment of the other lymphocyte, for example the T regulatory cell and CD8 T cell. So let's begin to understand the T cell play a key role in orchestrating pathogen specific adaptive immune responses. During primary infection, naive T cells recognize antigenic peptides, and this is the basically presented on major histocompatibility complex MSC class 2 molecules via their T cell receptor, and leading to their activation differentiation into effector T cells over the course of 1 to 2 weeks. Effector T cell means the different type of T cell which that is combat the inflammation site or pathogenic site. So that lead to proliferation and transcriptional, epigenetic and metabolic changes will be occur. So after clearance of pathogen, 90% T cell death by apoptosis. So why? Because no need to more killing of the pathogen. So we need to clear that through apoptosis. But important thing is that the five we will discuss. So the 90% cell death by apoptosis after clearance of the pathogen in our body, while the 5 to 10% differentiate into memory T cell such as central memory, effector memory, tissues resistant memory for quick response again when infection will be arrive in our body. Infection cancer can be infection simple or inflammation. So the naive T cell basically bind with the dendritic cell after the antigen representing cell to bind with the MSC class 2 molecule with the T cell receptor, and the inhibitory domain or inhibitory ligand, which that is also bind for inhibition of the naive T cell proliferation. So in this way the naive T cell proliferation inhibitor ligand is the PD-1. So let's begin to understand what is the PD-1, and first of all we will understand about the antigen presenting cell interaction with the naive T cell. So the synapse is we can say that is the here, and here you can see the antigen presenting cell which represent the antigen. Antigen, what is the antigen? Basically the antigen can be a pathogenic part after the endocytosed through a dendritic cell. So here is a T cell and upper part is the synapse which that is the cellular gap, two cells gap, between the antigen presenting cell as well as the T cell, naive T cell. So here the MSC class 2 molecule are present in the antigen representing cell, which show the T cell receptor is the microcluster formation degradation, and in this way you can see the T cell receptor complex formation, and this T cell receptor complex formation will be in this way the LCK will be bind with the T cell receptor complex, and in this way the cytoplasmic domain is the T cell LCK, and in this way here you can see this is the PIP2. This PIP2 is the phosphoinositide diphosphate, which that is a part of plasma membrane, and this is separate through a LCK activation by phosphorylation to activate the ZAP70, and this ZAP70 is activate the phospholipase enzyme C gamma. This will be breakdown of the PIP2 into the IP3 and diacylglycerol, and this diacylglycerol and IP3 which that is separate, and in this way diacylglycerol are present in the cytoplasmic membrane, but I am drawing here is just for understanding. So here you can see the IP3 will stimulate the calcium efflux from the endoplasmic reticulum, and the calcium increase into the cytoplasm will lead to stimulate the calcinurium for cascade of phosphorylation to NFATC1. This is phosphorylated NFATC1. This is bound with the DNA. After the binding with the DNA, the NFATC1 is phosphorylated and is used for the proliferation and differentiation and the recruitment through a production of cytokines. So in this way, basically you can see here is the nucleus contain DNA, and the specific gene bind with the specific NFATC1. So I hope you make sense about that. Proliferation of the naive T cell into the basically CD8 and CD4 and T regulatory cell. For example, cytotoxic T cell and T helper cell. So on the other hand, here is the LCK1 is also stimulate the PI3K pathway, which in this way the PI3K pathway will stimulate the AKT1 pathway, and in this way the AKT1 pathway stimulate the mTOR pathway, and this mTOR is used for the cell growth, cell proliferation and cell differentiation as well as the cytokine production. In this way all thing is due to the cell growth, etc., and cell differentiation also. So the after the phosphorylation of the mTOR will bind with the gene bind with the FOXO1. In this way the FOXO1 will be phosphorylated to detachment from the DNA and activated the protein. This activated the protein through expression of the PDCD1 gene. This PDCD1 gene, basically the FOXO1 is phosphorylated and go into the cytoplasm. And in this way the 40 and 3 types is the FOXO1. In this way you can see the expression of the PDCD1 will be increased, and this PDCD1 gene is used for the transcription to the translation of the promotion of cell proliferation and differentiation. In this way the CD4 and CD8 cell will be produced. So I hope you make sense. So next we will understand about the inhibitory ligand. This inhibitory ligand is known as the PD1. This PD1 is known as the programmed death 1 domain. So let's begin to understand for understanding of the HIV infection and why the T cell will not differentiate during the HIV virus. So in this way the inhibitory ligand will be bind, and in this way you can see when the infected cell of the T lymphocyte will be infected, and the T lymphocyte during the infected will be inhibit the proliferation. Why this proliferation inhibit? Because the programmed death 1 domain during HIV infection expression on HIV specific CD8 T cells positively correlates with high viral load, impairment of CD8 positive T cells, and functions disease progression and reduce CD4 T cell count. So the CD4 T cell count. So here you can see the PD-1 molecule is the basic inhibitor. In this way, it is intracellular domain. You can see the immunoreceptor tyrosine based inhibitory motif bind and recruit the immunoreceptor tyrosine based switched motif. Basically, the two domain, which that is recruit the SHP2 molecule, SHP2 protein, and this SHP2 protein will bind with the second domain of the intracellular inhibitory part of the PD-1. So the PD-1 can be inhibited by the antiretroviral therapy. So in this way, here is the SHP2 will be recruit and bind with the immunoreceptor tyrosine based switch motif, and in this way the SHP2 will be activated. After the activation, in this way, after the activation is the CBLB will be activated, and in this way the CBLB is activated to inhibit the T cell receptor complex, and in this way when the T cell receptor complex will be inhibited, so the proliferation of the T cell will be also inhibited. So in this way the proliferation will be inhibited and the pathogen associated pattern or the HIV infected cells will be not killed by the cytotoxic T cell due to the CD8 will be inhibited. So in this way the HIV virus is basically inhibit, you can see, and in this way here is the SHP2 will be also inhibit the DAG to Merck to ERK pathway. In this way it is all proliferative pathway, and here is the SKP2 is the inhibit by the SHP2, and it is the basically inhibit the CDK due to the inhibition of the SKP2. And on the other hand, the mTOR pathway is ultimately inhibited due to the PI3K pathway inhibition and AKT1 pathway inhibition through a SHP2. And in this way, the SKP2 inhibited to inhibit the CDK and the activation of the BATF, and which that is used for the inhibition of cell proliferation. So the cell differentiation and proliferation will not occur. So the mTOR will be inhibited. In this way the CDK will be inhibited. So in this way all things will be inhibited due to the SHP2, which that's all thing is used for the proliferation and differentiation through a PI3K pathway, AKT pathway, mTOR, as well as the calcineurin pathway. So let's begin to understand the HIV controller. If we will discuss about the controller, so the CD8 will be bind with the infected cell, and after the binding with the infected cell, the perforin granzyme produced, as well as the proliferation of the CD8 T cell also. So the structurally constrained HIV peptide if. But in the case of HIV progressor, in this way the progression of the HIV will be occur how? We will discuss about that. So in this way the HIV progressor is we will discuss. So the interferon gamma is recruit the CD8. So in this way the important thing is that basically I want to say that here is the after the binding with the inhibitory ligand and exhaustion of the cell, and in this way the inhibitory receptor will bind with the MSC class 2 molecule with the infected cell. So in this way the infected cell it interferon gamma will produce. So in this way the inhibitory ligand bind with the inhibitory receptor PD-1, and in this way the inhibition will be occur and the interferon gamma will not produce, and in this way the CD8 recruitment will not occur. So in this way the infection progression as well as the mutated HIV peptide will be occur, And in this way, mutated HIV peptide is occur due to the escaping of the HIV for the infection to another T cell. But this mutated HIV peptide will be occurred due to the lack of HIV after the infection. So the after the infection again recovery. So it is due to the PD-1 molecule, that is the inhibitory molecule, will inhibit the mTOR pathway and mTOR pathway and ERK pathway, as well as the CDK and SKP2 pathway inhibition. So in this way, the cell proliferation into CD8 cell, T regulatory cell, and produce the chemokines and cytokines for recruitment of the other innate lymphocyte and adaptive immune cells like T cell, B cell will not produce. And this all things is related to the immune response. So when infected cell of the HIV infected cell will not give the response, so in this way the infection will be progress.